Okay, hi everybody. I'm Lincoln Stein. I work here at OACR, and I'm one of the principal investigators on the Reactome project. And Nia, can you show me how to advance?
Yeah, you can just use the arrow keys.
Okay,
Theoretically.
in theory. That's not doing nothing.
Yeah, there you go. Just need it to select the screen.
Oh, select the screen.
Yes.
Okay, good. So today we're going to talk about pathway and network analysis, and I'll give you a little bit of a soup to nuts introduction to the various basic and advanced forms of this work. So we're going to talk about the principles of pathway and network analysis, where the data comes from, how you can get it, the various differing approaches to using data analysis and visualization of this data, and some applications of pathway enrichment analysis to whet your appetites. So the main reason people want to do pathway analysis, as I'm sure you know at this point, is the dramatic data reduction in the size of the data set. Instead of trying to find patterns in thousands of genes, you have a small number of pathways, maybe numbering in the dozens. By looking at changes at the pathway level, you're increasing your statistical power by reducing the number of hypotheses that you're performing. You may be able to find meaning in the long tails of infrequent changes in the data set. Here I use the example of rare cancer mutations, but it could be anything like genes which are infrequently altered in your system. And the real value of pathway analysis is that it gives you ready-made biological stories. So it tells you which biological functions have been altered in your system, and lets you develop hypotheses that you can then test. And it's a lot easier than dealing with hundreds of poorly annotated genes with obscure accession numbers and names. So this is a very broad pathway network analysis is a very broad term, encompasses many actually very different techniques. Here I'm using it to describe any analytic technique that uses biological pathway or molecular network information to gain insights into a experimental system. It's rapidly evolving, and there are many approaches. And in fact, over the last couple of months, I had to add a new section to this talk because of a whole new class of AI based analytic techniques. So basically, you only need two types of ingredients to do pathway network analysis. You need a high throughput biological data set, such as a list of altered genes, proteins, or RNAs. It can come from proteomics, it can come from kinase screens, it can come from RNA-seq or single-cell RNA techniques, but you need a large sample size and hopefully a large number of perturbations of the system. And then you need a framework of annotated pathways and/or gene networks to work on. Now, the basic difference between a pathway and a network is that pathways are biological descriptions of the biology of a series of reactions that occur in a cell or between cells, and are the kind of the fully annotated biochemical pathways that people and models that people publish, in which you have a series of reactions which have things that go in, inputs, which can be proteins or RNAs or lipids or whatnot, and a series of outputs, modified proteins, So a kinase reaction will take as an input a protein and an ATP, and its output will be ADP and a phosphorylated protein. These are great at telling biological stories, but it's hard to analyze over because it's very heterogeneous. You're dealing with many different macromolecules that are interacting with in complex ways. So network analysis is networks are a simplified view of the interactions among macromolecules in the body. They have more of a genetic view, and so here we're looking at the EGFR signaling, the first few steps of ligand binding to the receptor. On the left, you see the pathway view, and the right, you see the network view. And you can see that in the network view, everything looks rather uniform, and there are two types of arcs. There are activators and inhibitors. And that's a very simple model that's been built from the more complex biochemical model on the left. So just as there are two different ways of representing pathways and networks, there are different types of databases. So the older ones are pathway databases, which are generally curated databases. They come out of the literature. Human beings have reviewed papers and have extracted biochemical reactions and put them into a database in a standardized format. They give you the biochemical view of biological processes. They capture cause and effect, and they have visualizations that we're familiar with, pathway diagrams. The disadvantages are this is very labor intensive, and science does not understand all of cell biology, obviously. And so the genome is covered sparsely. At best, pathway databases cover maybe half of the genome, probably not even that much. And because human beings are involved and it's coming out of the scientific literature, the definition of what a pathway is will vary from one database to the next, depending on our history and arbitrary decisions that curators and people doing the experiments have made. So, where does the EGFR pathway begin and end? That's very subjective. So the two biggest accessible databases, non-commercial databases, are Reactome and KEGG, which I'll talk about in a moment. And they both have similar data models. They're reaction network databases, which describe pathways as a series of linked reactions. I sort of described this earlier. It's the fundamental unit of these databases is the reaction. Reactions have inputs, which can be any macromolecule or small molecule for that matter. And then it has a series of outputs. And sometimes the inputs and outputs are related by a chemical modification, a phosphorylation reaction, a cleavage reaction, a binding reaction, or sometimes it can be more subtle, such as translocation from one cellular compartment to another. So, ATP outside the cell is not the same as ATP inside the cell. And then there are regulators which up and down regulate the reaction. The older of these two is KEGG, a long-standing, I think it's a 30-year project out of the University of Kyoto. It's a really large library of genomes, genes, proteins, pathways, and chemical compounds across hundreds of different species. It features a resource called KEGG Pathway, which is a collection of manually drawn, quite attractive pathway maps that represent various pathways in metabolism, cellular processes, organismal systems, human diseases, and drug development. As you kind of see from this list, it's kind of arbitrary chapters, but they work well. The KEG used to be completely open access, but in the last 10 years or so, it's becoming increasingly hard to access the underlying data. You basically now require an institutional subscription to actually download the data set and do analysis across it. What you can do with it now is browse through it and look at the maps. So here's what a typical KEG map looks like, representing the cell cycle. One of the features of this is that each of the molecular interactors is described using a uniprot number, so it's well known, and each arc is described using an EC, enzyme catalytic number. The project I work on is called Reactome. This is a 20-year-old project, so it's not quite as old as KEG. It is completely open source and open access. In fact, everything in Reactome is in the public domain. So that's made it widely used both in academia and industry because there's no licensing restrictions attached to it. It's curated by a group of curators located in Canada, the USA, and the UK, and sometimes in other parts of the world. We had a curator in Vietnam for many years. And it focuses very strongly on human pathways. It doesn't attempt to cover all organisms. It spans everything from metabolism to cell differentiation. We have rigorous curation standards. Everything in the database has a citation in the primary literature, and we have a review cycle where each curator's pathway gets reviewed by outside experts, PIs and senior postdocs, and gets updated and brought up to date. We do cross-references to other informatics databases, so when you see a protein or a gene, you know what it is unambiguously. And we provide a series of visualization and analysis tools that I'll talk about. So here is what the browser interface to Reactome looks like. There's a search function that lets you find pathways by name, or you can search for a gene and find out what pathways it participates in. It has a series of, I don't know if my cursor shows up here. No, okay. It has, at the upper level, there's a text description, which you probably can't read, and a nicely drawn picture of the pathway. And then as you drill in, you get increasingly detailed until you get down to the biochemical pathway like description, and you can continue to download this information in a variety of formats and load them into analytic tools. So any questions about that's what pathway databases are basically look like. They use internally both KEG and Reactome and other pathway databases use a series of industry standard formats for describing pathways, including BioPACs and CellML, which I will not get into, but they are text representations.